Now that we've covered the basics of how your genome is in charge of the growth and development and replication of cells and the DNA that is contained within them, we can delve into the genetic basis of cancer, in that cancer itself is 100% a genetic disease. So, cancer is this really broadly applicable term that applies to many different diseases that manifest themselves in many different ways. Different parts of the body, different tissues, different life stages of the individual. And what's even more remarkable is that two individuals might have the exact same cancer, whether it be breast cancer or colon cancer or skin cancer, and the mutations that caused them to have that disease could be completely different. So, this is why cancer is such a complex disease to treat and try to figure out a way to beat. Okay? So, cancers all have two common characteristics, though. And those characteristics are this abnormal cell growth. So, what a cancer is at its heart is this uncontrollably dividing group of cells that manifest themselves into what we commonly refer to as a tumor. Okay? And so, you have learned about the cell cycle and you understand the things that happen before mitosis happens. And we're going to talk about in a bit more detail how that's regulated today and how that regulation is lost in cancerous growth. Cancer cells also have the ability to invade other parts of the body. Now, this is also something that we don't necessarily think about as part of cancer, but when you find out that someone has been biopsied for a tumor, right? You find out, let's say, a loved one just came back from the doctor, they had a biopsy, they're waiting on the results. Okay? So when those results come back, they're going to tell you this is a tumor, but they will come back with one or two terms, benign or malignant, and that tells you whether this tumor is staying where it is and just growing uncontrollably there, in which case we cut it out and we're good to go, assuming you're able to cut it out. Or are those cells that are dividing, not only dividing there, but also spreading to other tissues in the body? Okay, so to be characterized as cancer, the cells have to have both of these characteristics: uncontrolled growth and the ability to move about the body. So, to underscore the dramatic changes to the genome that are associated with cancerous growth, here is an example of a karyotype of a human male, 23 pairs of chromosomes. Those chromosomes have been dyed so that you can identify which chromosome is which, and everything looks pretty kosher here, right? Nothing terribly out of order, no mixed up colors, no real different sizes, and they have two of everything. Here is a karyotype that is typical of a cancer cell. And as you can see, there is a whole lot of stuff going on here. These cells have 3 or even 4 of some of the chromosomes. You can see for chromosome 8, they have 4 copies. For chromosome 10, there are 3 copies. Chromosomes aren't all the same size. Look at chromosome 2. One of them has an extra bit of something else attached to it. I don't know what's going on with that chromosome 4 on the right. Chromosome 10, the one in the middle, the third one, the extra, it's only half of a chromosome 10. Look at the 18s, look at the 13s. There are so many different mutations in these cells, and this is part of the reason that cancer is such a difficult disease to treat, because there are so many things going wrong in these cells. Okay? So let's first talk about the fact that these cells are growing uncontrollably. This is how we receive these tumors, through this uncontrolled division. Now, this uncontrolled growth on its own isn't necessarily problematic. Okay, an uncontrollable group of cells is simply a tumor. If it's not moving about the body, it is said to be benign, and that's not necessarily a problem. If I have a benign tumor on my arm, it can be cut out. Now, if I have a benign tumor in my brain, that's more problematic. Getting it out of there could be problematic. But for the most part, these benign tumors are easily treatable, and they're not of great concern because they haven't spread around the body. Okay? Now, when cells of a benign tumor gain this ability to spread or metastasize, then they are no longer considered benign, but they are malignant. They are capable of moving about the body and popping up in other tissues. This is what your oncologist is most concerned about when you're diagnosed with a form of cancer. Sorry, I think I accidentally hit my mouse pad, so I stopped recording for a second. So this is what your oncologist is worried about, is the spread of these tumors throughout the body. And so understanding whether or not these tumors have, these uncontrollably dividing cells have spread around the body, that is of chief concern to your oncologist. Okay. Now, cancer cells are clonal. This is interesting. Okay, so if you develop colon cancer, for example, it might not be the colon where those cells are first noticed, where this uncontrolled cellular division is picked up. So, for example, my father was diagnosed with colon cancer. The tumors were first discovered in the liver. Okay, and so, but because of the characteristics of those cells, that led the doctors to immediately trace back to the colon and identify the original tumor that had set the whole thing in motion. So when you have a metastasizing cancer, tumors can pop up in many different tissues of the body, but they are there because of that metastasis, because of that movement around the body. They are growing uncontrollably in each of those tumors for the same reason, because they have the same mutations. It's not that I develop colon cancer, I have cells uncontrollably dividing in my colon, and then the same thing happens independently in my liver or my brain or my kidneys or wherever. Okay, so this is substantiated by the fact that all cancerous cells in a woman that has a metastasizing cancer, they all have the same inactivated X. Remember back to our discussion of the Lion hypothesis and X inactivation. So there are some really consistent patterns that make clear the reason that these tumors are popping up throughout the body is not because they're independently arising, these mutations and this uncontrolled growth, but because they are actually spreading. Now, it probably goes without saying that cancer requires multiple mutations, and as you saw from that karyotype, multiple mutations there are. Okay, but this explains why cancer is age-related, right? It's not a disease that you see terribly often in very young individuals. Okay, cancer is most commonly encountered in older individuals, and this is because of the need for the accumulation of multiple mutations into genes that contribute to the regulation of the cell cycle. I want you to think about this very carefully as we get into the finer details of how cancerous mutations affect the cell cycle and prevent the cell from regulating its own division, okay? Because it's that uncontrolled division, uncontrolled growth of these tumors that we have these problems, okay? Now, it takes a lot of checkpoints, a lot of important genes that would normally keep this division in check and in order. A lot of those need to be mutated in order for us to see this uncontrolled growth. And so what you've got to realize is that this all stems from a single cell in which genes 1, 2, 3, 7, 9, and 11, all of them had a mutation that made it so they didn't function properly anymore. And once all of those perfect combination mutations happen, then that cell has no regulation of its division any longer. Okay? So once these cells reach this point, they have enough mutations that they are dividing uncontrollably. They are not repairing their DNA any longer, and they become more and more tumorigenic, meaning that they divide even more rapidly, and more malignant, so they spread more and more efficiently throughout the body. Okay? So you suffer many, many mutations. All of those mutations are random. Some of them might be induced. Let's say you go out to the beach and you don't wear sunscreen. Some of them are just random chance. You could lay in bed all day under the covers and you're still going to suffer a good number of mutations. Okay? So your DNA actually suffers somewhere on the order of 10,000 mutations every day. Now, this is a shocking number, but as we've discussed already, we have mechanisms that are capable of repairing these. Okay? So you have lots of mutations every day, and you fix damn near every one of them. The problem is you're never going to get every single one every day. Okay? But even if it's 99.9% effective, okay, you are still left with a couple of mutations every single day of your life. Now you've got millions of cells, okay, but accumulating mutations every single day, you're going to start accumulating mutations in the same cells that already had mutations. And by the time you're in your 60s, 70s, 80s, 90s, you have cells that have suffered many mutations, some of which can combine to form that perfect storm of a cell that can no longer control its or regulate its division or maintain its self-identity and prevent itself from metastasizing. So cancer cells are characterized by lots and lots of mutations, and this is exacerbated by an enhanced mutational rate, because most often one of the mutations that's necessary within a cell that becomes cancerous is mutating and inactivating the very genes that are responsible for repairing errors. Okay? Once you lose the ability to repair errors, every error that happens, all 10,000 of those every day, are going to be kept. Okay? Cancer cells are characterized by these chromosomal abnormalities that you saw in that karyotype and general genomic instability. So there's all sorts of crazy stuff going on with respect to the balance of numbers of chromosomes in the genome. And so all of this is linked to a lack of repair, and this is consistent with what we learned about the genetic disorder XP, where those individuals are born without the ability to repair DNA mutations, and they have a highly increased risk, 2,000-fold risk of cancer relative to someone that is capable of fixing mutations or repairing genomic issues. So this cancerous karyotype we saw should make it clear that these types of instabilities within the genome, like duplication, loss, translocation of chromosomes, are quite common. But it's interesting in that some of these might be used to identify or characterize the specific type of cancer, and in some cases may even inform the way in which that cancer might best be battled using drugs and other pharmaceuticals. Okay, so one example of this is chronic myelogenous leukemia, and this is a cancer that is characterized by a non-reciprocal translocation between chromosome 9 and chromosome 22. So a little bit of 22 gets put on 9, and a little bit of 9 gets put on 22, and it's not exactly the same bits. And what it does is it creates on chromosome 22 this fused gene, where instead of just having the BCR gene on 22 and the CABL gene on chromosome 9, there's this fusion of BCR and ABL. Now, normally ABL transmits growth factors to the cell. And so ABL has a very important role. It hangs out and waits for the cell to receive external signals that tell that cell to divide. So ABL is responsible for taking that information to the nucleus and telling the cell, or giving to the cell the information that it should be dividing. Okay? But with this mutant protein created by fusion of the BCR and ABL genes, that protein does not transmit this signal faithfully, and it constantly stimulates the cell to grow and divide. It acts as if the cell is constantly receiving signals to divide from outside the cell. The nucleus, or the brain of the cell, if you will, has no idea that there really has been no signal suggesting that division is needed or necessary, and so it has no reason to disbelieve this protein that is initiating mitosis. Right. So these are the types of mutations that are responsible for cancerous growth of cells. And again, as I mentioned, the XP condition, this phenotype can be the result of mutation to any one of 7 different genes involved in DNA repair. So remember, I said 2 people could have the same cancer, but for very different reasons. This condition, xenoderma pigmentosa, is very much the same thing in that there are 7 different genes that can be broken or mutated that cause these individuals to have no ability to actually repair their mutations to their DNA, and they are thus more likely to develop cancer. So what we're dealing with in cancer is an actual alteration of the cell cycle in which cells grow and divide uncontrollably. Now, the cell cycle is different in different tissues. Some of your cells are constantly growing and dividing. Some of them don't grow and divide as you reach adulthood, okay? But there are lots of genes that control the cell cycle and whether or not a cell leaves interphase and enters mitosis. Okay, so you will remember this figure from the first week of class when we discussed mitosis. Many cells exist, most cells exist the majority of their lives in G1 and or G0. So G0 is basically you're just doing your thing as a cell and you will never divide. Some cells might jump back into G1 as they prepare for division and entering the S phase, okay? So what I've got here in red are 3 different checkpoints that we haven't talked about yet. So you've seen everything here except what's in red and these 3 checkpoints. So you'll see that there's a checkpoint between G1 and S, there's a checkpoint at the end of G2, and there's a checkpoint in the middle of mitosis. And I just want you to recognize from these checkpoints that there are a number of periods during the cell cycle when cells absolutely must self-examine to ensure that proceeding to the next step in the cell cycle would not potentially result in some catastrophic change leading to potential problems downstream. So let's first start with the G1S checkpoint, okay? Now as a cell goes to enter the S phase, which is where DNA replication takes place, it's only doing that if it has been told to divide, and in this checkpoint period, the cell must self-examine to make sure it is large enough to be broken into 2 to undergo division, and it also, as it's about to replicate the DNA, it must also determine whether or not the DNA is of sufficient quality or has the DNA been degraded or mutated by, say, exposure to UV light. Okay? If the integrity of the DNA is low, if something's wrong, you don't want to duplicate it in the S phase. So the intention of the G1S checkpoint is to make sure the cell's ready to divide and the DNA is ready to divide, or replicate, be duplicated. After the DNA is replicated in the S phase, the cell in G2 prepares for division in mitosis. And at the end of G2, there's a checkpoint whereby the cell monitors the DNA, makes sure that the replication was effective and efficient and accurate, and makes certain that the DNA is not damaged. Once the DNA is determined to be good and undamaged, the cell may enter mitosis, during which there is one final checkpoint, okay? And the M checkpoint, I would think of this as the metaphase checkpoint. What the cell is doing there is monitoring spindle formation and the attachment of kinetochores. Remember, this is how the cell breaks apart identical sister chromatids and pulls them in opposite directions into the two daughter cells. If those kinetochores are not properly connected on both sides, if you only have a connection on one side, you might end up with trisomy, right? You're going to pull both of those to one side if only one half of the centromere is connected to a spindle fiber. And so it's critical that the cell ensure that there are connections on both sides of every centromere such that those replicated chromosomes will be pulled apart in opposite directions to go into these two daughter cells. Okay, so this is how the cell regulates itself. Now, how does it know when to divide? The cell is generally responding to external stimuli, so the transmission of growth signals from outside the cell hit the cell surface, and that information is then carried to the nucleus. This is called signal transduction. Okay, so signal transduction can induce a cell that is in G0 to re-enter the cell cycle, okay, right here. So you see there's this decision of the cell to re-enter the cell cycle. And once that signal transduction takes place, we're going into the latter part of G1, hitting that checkpoint, S, G2, the last checkpoint after G2, and then the metaphase checkpoint. Now, during the cell cycle, there are lots of new proteins and other enzymes that need to be produced so that the cell can replicate effectively. So one example is DNA polymerase, right? DNA polymerase is used for the replication of chromosomes. Does a cell in G0 need DNA polymerase? Certainly not. Okay, DNA polymerase is only needed when you're making copies of the DNA. That's the S phase. If the cell never goes into the S phase, it doesn't need polymerase. Okay, so once the cell is induced to undergo mitosis, there are a lot of new proteins and enzymes that need to be made. Okay, and so there are things that get this started, and these are cyclins and CDKs. CDK stands for cyclin-dependent kinase. So we've got cyclins and kinases, the kinases being dependent on the cyclins, okay? So in addition to these cell cycle checkpoints where things are being examined for integrity and size and strength and proper attachment, we also have the regulation of the production of cyclin and CDKs that contributes to whether or not the cell will enter the cell cycle, okay? So here's a graph of the abundance of these different cyclins that exist. And these are things that, again, they initiate the production of polymerase or ligase or helicase, any of those things, breaking down the nucleus in mitosis. All of these are things that have to happen, but you don't want them to happen all the time. Only if the cell is about to divide do you want all of these things. And so you can see that cyclin E, cyclin D2, cyclin A, those are not always around in the cell. They are only around very certain periods because, for example, D2 is a cyclin that is part of or drives the replication of DNA. And so you need lots of D2 as you enter the S phase. Once you begin the S phase, you no longer need to keep producing D2 and its production is shut off. So think about the regulation of gene expression that's involved in the maintenance of the cell cycle, where the cell is controlling when and if these cyclins are produced. The cyclins, once they're produced, they induce the production of other things like ligase and polymerase, and then their role is done once that portion of the cell cycle has been passed. Now there's a term that I want to introduce you to that you may have heard before, and this is apoptosis. Okay, and lots of people pronounce this different ways, but what this is programmed cell death. So if a cell is incapable of doing what it should do, if it is incapable of repairing DNA, or it identifies such catastrophic errors in the genome or the attachment of spindle fibers, and it can't be fixed, the cell would rather die than contribute to a legacy of mutated, maladaptive, poorly functioning cells. Okay? And so there are proteins that are involved in regulating whether or not the cell enters apoptosis or not. So if things can't be fixed, that cell needs to be destroyed. Okay? And so the process of apoptosis, you don't need to memorize all of these different steps, but it's basically a cell shrinking upon itself, destroying its cellular membrane, and just dissolving into small little pieces. Okay? So this is what we do with a cell that is so far off the rails that it cannot be repaired. Okay? So this becomes relevant when we start talking about some of the ways in which the cell cycle is regulated, because you think about the cell entering each of these checkpoints, and each of these checkpoints is where something is reviewed and repaired. But there's got to be an option for the repair isn't working. And this is it. This is the offshoot of the cell cycle when a checkpoint can't be passed. Okay, so when we think about cancer, there are two basic types of mutations. There are mutations to regions that are characterized as proto-oncogenes, and then there are the mutations that are to regions that would be considered tumor suppressor genes. And we're going to go over each of these, but it's critical that you understand the distinction between these two. Proto-oncogenes are those that stimulate the expression of other genes. So the product, the gene product, the protein produced by expression of a proto-oncogene is a protein that stimulates the expression of other genes necessary for cell division. Okay? And so a great example of a proto-oncogene would be a signal transduction factor, some protein that carries that information from outside the cell to the nucleus to tell that cell, hey, we've received a signal, it's time to divide. This is the type of gene product that you get from a proto-oncogene. Okay? So they are regulators of the cell cycle. They're the ones that shepherd the cell through the process, telling it when to make ligase, when to make helicase, when to start doing all of these things. When these cells are mutated, the cells cannot be controlled. Okay? A mutant form of a proto-oncogene is an oncogene. Okay? So this was a gene whose gene product is responsible for stimulating the expression of other genes and making the cell cycle happen. Okay? But when you mutate that, you end up with an oncogene product, and that's problematic. Now I'm going to go through an example of both types of cancerous genes, and this will make the distinction between them much more clear. A tumor suppressor gene is one that regulates the cell cycle checkpoints. And if that checkpoint cannot be passed, if the damage to the cell, the problems within the cell are too difficult to repair, then the tumor suppressor gene product will initiate apoptosis. Now, in order for this to happen, you need 2 copies of the mutated gene. Okay, so this is a tumor suppressor gene is what we call a loss of function mutation, in that the tumor suppressor gene is no longer doing what it needs to do, is not initiating apoptosis. And the reason I say you need two copies is, think about this. Think about a protein whose job it is to initiate apoptosis if things in the cell are really, really bad. Okay? If your mom gave you a copy and your dad gave you a copy of that gene, you express that gene, you have proteins that result from the expression of those gene copies that you got from mom and dad. If you have a mutation to one of them, okay, let's say there's a mutation to the copy that your mother gave you, and you produce a non-functional protein that will initiate apoptosis, meaning it can't initiate apoptosis if stuff's really bad. The thing is, you still have a functional protein gene product from the gene copy that your father gave you. And so you will be able to initiate apoptosis despite the fact that one of your copies of this gene is mutated, okay? In order for you to lose the function of this gene in your genome altogether, you must have a mutation that, to your mother's copy, that results in the production of a non-functional protein, and you must also have a mutation to the copy that your father gave you that also produces a non-functional copy of the protein. Neither of those is capable of initiating apoptosis, and you have therefore lost the function of this gene. But you have to have a mutation to both of them, okay? Another example of why cancers are associated with age most frequently, because it's going to take a while before you have a mutation to the same gene twice in one cell. So that is the type of gene that is responsible for maintaining the integrity of these cell cycle checkpoints. So let's go through two examples of, or an example of both a proto-oncogene and a tumor suppressor gene. The RAS proto-oncogene, this is just one gene in your body, but the RAS proto-oncogene is one that is mutated quite commonly in cancers. Now, to be clear, to remind you, just mutating this is not going to give you cancer, okay? It's the combination of a mutation to RAS and a couple other things that breaks enough of these checkpoints and the redundant abilities of the cell to destroy itself or repair itself that results in cancerous growth. But in the changes that we see to the RAS proto-oncogene and that we will discuss here, it should be clear to you how a mutation to this gene could lead to uncontrolled growth. And so here's what RAS does most of the time. It's right there in the middle, okay? As a proto-oncogene, it is responsible for transmitting the signal from the cell surface to the nucleus, telling the nucleus it's time to divide. Go ahead and replicate that DNA, and we're going to divide. We're going to become two cells here in a bit. You'll see this whole process is illustrated starting with a growth factor that interacts with a receptor on the cell surface. When that growth factor does this, it initiates a cascade of events within the cell, starting with this phosphorylation of RAS, okay? So RAS goes from this inactive state, it's not telling the cell to do anything, okay, to an active RAS state that then will initiate this daisy chain of events, okay? It sends a signal to RAF, which signals MEK, which signals MAC kinase, which initiates the distribution of these transcription factors within the nucleus that begin the process, okay? So because it is sending cells from the cell membrane to the nucleus, telling the cell to divide, when mutated, a mutant form of RAS, instead of doing this transiently for a while when that growth factor is sitting in the cell surface receptor, and then once that growth factor is no longer there, it reverts back to the inactive state. The mutant forms of RAS are incapable of reverting back to their inactive state. And so once phosphorylated, once they receive that signal, once turned on, that cell will never turn off again. RAS will never stop telling that nucleus it is time to divide. Okay, and this is hugely problematic. And so, unlike tumor suppressor genes, where you have to have both copies of the gene within the cell mutated and broken, in a proto-oncogene, you need only one copy of this gene to run off the rails, to be mutated, to not function properly, and get uncontrolled growth. Okay? Imagine in this cell, if only half of your RAS proteins are mutated and incapable of becoming inactive. So once the cell signal, the growth factor signal goes away, half of your RAS reverts back to that inactive state. But it doesn't matter, because the other half will never revert to the inactive state. It will continue sending this division signal to the nucleus. And for that reason, you only need one of the copies of that gene in your genome to have been mutated to suffer this uncontrolled growth. Okay. All right. Now, P53 is an example of a tumor suppressor gene. Okay. This is a gene that is mutated in more than 50% of cancers. So this is a very common player in cancers that we see in humans. So the protein P53 is a transcription factor. Okay, and what that means is it's something that facilitates the transcription of things that are needed for the S phase. Think polymerase, primase, all those things you need to replicate the DNA. Okay. Now, most of the time, it's hanging out, teamed up, stuck to MDM2. This is another protein that binds P53 and keeps it busy, keeps it from stimulating the cell cycle. Okay? So it basically, P53 is around all the time, but it's almost always targeted for destruction. Okay? It's not to be used, it's just there just in case. So when P53 is available and doing its thing, it is going to stop the cell cycle or initiate apoptosis. Okay? So it's this transcription factor that's inhibited by MDM2. If it's not inhibited, it's going to stop the cell cycle or initiate apoptosis. So it's regulating whether or not the cell is going to divide. And here's a schematic that shows you when P53 gets activated and why. Okay? So in the cell, if there is DNA damage to the genetic material in a cell, the P53 gets activated. It's the repairman of the cell. So you can see that when the DNA is damaged, P53 and MDM2 no longer hang out together. Okay? P53 gets to go out and do its thing, and that thing is stop the cell cycle, make the things that will repair the DNA, and then throw the cell cycle back into play. Okay, get it back going. Alternatively, if P53 is incapable of fixing things once the cell cycle has been halted, it will push the cell to apoptosis. Okay, and so what this means is P53 is kind of like the policeman of the cell. It makes sure everything is okay, it fixes things that are wrong, and if not, it will ensure that the cell does not go on to hurt the overall body of the organism. Okay? So, when we think about cancers, we think not only about the uncontrolled growth, but we need to think about the movement of these cells throughout the body. And so what you need to recognize is that between all of your distinct tissues, your liver from your stomach, from your kidneys, okay, there's an extracellular matrix and these basal lamina that separate all these different types of tissues. What cancer cells have to be able to do is degrade these and move through them from one organ system to the next. We're not going to get into the genetics of how that happens, but I just want you to understand the mechanism and the basis of metastasis. Okay, so one thing I want to touch on here is something that a couple of you asked me about in office hours one day, and that is the heritability of cancer. How can you inherit a predisposition to cancer? Well, a small but significant number of cancers are actually heritable. And this is the important point. You generally inherit a single mutant cancerous version, okay? And you have to undergo what is called a loss of heterozygosity, okay? Now, this should make sense to you based on the systems that we just described, proto-oncogenes and tumor suppressor genes, okay? In one of those systems, and I encourage you to make sure that you understand which is which, you need both copies of the gene to be non-functional, to be broken for you to have cancerous growth, okay? So what that means is you can inherit from one of your parents a non-functional, broken copy of that gene, but you got a functional copy from the other parent. So basically, your entire body is heterozygous for that gene. Every cell in your body, you have one functional copy and one broken copy. What that means is every cell in your body is one mutation away from losing the function of that gene completely. Whereas someone that is homozygous for the functional copy of the gene, they have to suffer 2 mutations in the same cell line to lose the function of that gene, okay? And this is exactly why cancers like breast cancer, we have genetic screening that can tell you, yes, okay, for this BRCA1 or BRCA2 gene, you are heterozygous, you inherited a non-functional copy from one parent. This makes you X% more likely to develop breast cancer than someone who is homozygous for a functional copy of this. So if you lose your heterozygosity, that means you lose that one functional copy in a cell, then that cell no longer has that ability to maintain the integrity of the cellular machinery and prevent it from entering mitosis, and you get uncontrolled growth. The text talks about the heritability of some cancers like colon cancer, and there's a continuum of colon cancer that goes from normal colon epithelium all the way to colon cancer. And in between are epithelia that kind of grow abnormally, they become benign, they become intermediate adenomas, then they start developing these other structures and metastasizing. It gets pretty bad. But you can see that you might be born with a mutation you might be born heterozygous for one of these genes, and it is the loss of function of that gene by a mutation later in life to one of these other loci like P53. If you lose the function of P53 through a loss of heterozygosity, then you end up with another of these boxes checked off that allows you to have this tumorigenic growth. So the last bit I want to talk about with respect to cancer is the contribution of viruses to cancers. And this is something we don't think about all that often, with the exception of one important example, okay? So viruses can come in and cause cancer because they might be retroviruses. They bring a bit of RNA into our bodies, and that RNA gets stuck into our DNA, okay? So remember, we learned about retroviruses and proviruses, and what they bring with them when they do this are these powerful enhancers and promoters, okay? These things can be very strongly expressed within our cells and alter our ability to regulate the cell cycle. And HPV is one of the perfect examples of this, okay? You are probably familiar with HPV. Many of you have probably received the vaccine for HPV. And that's an important one because HPV is associated with 90% of anal and cervical cancers, 60% of penile cancers, and 65% of oral cancers. Now, for a long time, this is a vaccine that people thought only young women had to get. Okay, so you can get this up to a relatively late age. I can't go get the vaccine, but you are still young enough to get the vaccine. But what it does is it prevents you from contracting HPV and having these increased risks of cancers. Now, where I was going was for the longest time, it was thought that only women needed this. Okay. But it turns out that men are increasingly developing high rates of cancers, particularly oral cancers associated with this. So if you know how HPV is transmitted, I will let you put together the dots, connect the dots on how males are developing oral cancer from exposure to HPV. But needless to say, the rate of oral cancer in men these days is exceeding the rate of oral cancer. The rate of oral cancer for men from HPV is exceeding the rate at which men are developing oral cancer due to tobacco use. Okay, that's dip, chew, cigarettes, all of it. Okay, so this is an epidemic. It is increasing in frequency, and men should absolutely get the HPV shot. And I know all of the men in this class are still of an age where they can still get the shot. So if you haven't gotten it, ask your parents. If you don't know, but seriously consider getting it. Because here's what HPV does. And we're going to focus primarily on the P53 molecule, this guardian of the cell that we spoke about, that prevents the cell from dividing when it shouldn't. When the DNA can't be repaired, it initiates apoptosis. Well, guess what? This integrated HPV, this gets stuck into your genomic DNA, and HPV produces these proteins E6 and E7. We haven't talked about RB, so I won't really talk about E7, but what E6 does is it binds to P53 and destroys it. You know what that means. If P53 is not there, P53 can't have the cell fix itself. It can't destroy the cell if things can't be fixed. When P53 is gone, you lose the tumor suppression activity of P53 and you get uncontrolled growth. Okay? All right, so that's the end of our chapter on cancer, and we've got one to go.